Hello, my name is Songdi. I'm a graduate student at the University of Manitoba. Today I will be talking about tree decomposition for the reconstruction of ancestral RNA sequences under multiple structures with base pairing and stacking constraints. Understanding evolutionary relationships and scenarios has been one of the main keys in the development of better sequence mutation models. The reconstruction of ancestral non-coding RNA sequences is particularly challenging due to the main conservation forces being applied to the structure rather than the sequence. Traditional methods, like Fitch or Sankoff, mainly consider the conservation of sequences. However, RNA families are known to favor the conservation of structures over sequences, so it can be used to guide the reconstruction of ancestral sequences. Astronomy 1.0 by Tremblay-Savard et al. Considered such structural constraints. This figure shows the motivation as well as the high-level idea of Astronomy 1.0. In the left figure, the red and blue areas can represent the ancestral sequence reconstruction with structure S and S'. It is believed that the common ancestor of two homologous families was able to fold into both structures. As we can see here, proceeding with a single target structure could introduce a bias to the results. For example, the red and the blue could each end up with a different set of ancestor sequences that is able to fold into only one of the structures. The species tree at right involves 4 species. It is assumed a duplication event at the root where 2 ncRNA families are created. Followed by a series of mutations, these families gradually became more focused on fitting one of the structures. And that is why, in the middle of the graph, we have a parameter g or gradient that controls the weight that is given to each structure depending on the depth. In this figure, 2 families with their structures are shown. We can see that for family 0, position 3 is paired with 5, but position 5 in family 1 is paired with 2. Depending on the structure, such a we call interdependent relationship could create a long path of positions. Each position on this chain would increase the time complexity by 4 since we have 4 possible bases to fill in. To simplify or limit the depth of the interdependency, Astronomy 1.0 would stop at one level, that is, it would look only at one paired position in the other structure for each position in the first structure. And that's the problem we try to solve in our study. Okay, now let's talk about our new algorithm. Before the reconstruction, we need to pre-process our data. Here is a small example of 2 structures S1 and S2. In the top right figure, the green edges connect base pairs in S1, while the yellow ones connect base pairs in S2. The black ones represent stacking relationships. Consider all edges in the figure. It is a graph that can be easily transformed into a tree decomposition, which is the method we apply to solve the interdependency mentioned in the previous slide. Here is a tree decomposition based on the networks in the previous slide. In the tree decomposition, each bag contains positions that are related, for example, base pairs and stackings. Before using the tree decomposition for cost calculation, we needed to be able to bound the time complexity, which in our case depends on the tree width of the tree. To optimize the tree decomposition or to reduce tree width, we applied a method called tree diet. It is a method proposed by Marchand et al. That can slim down the tree decomposition to a prescribed tree width by removing the minimal set of edges. So after preprocessing, here comes our main algorithm. Our new algorithm, Astronomy 2.0, consists of 3 steps. A bottom-up step that performs a post-order traversal to the species tree. During the process, the minimal cost for each possible set of nucleotides in each bag of the tree decomposition is calculated. A middle step that links the minimal cost matrices for both families at the root of the phylogeny. Then it's a top-down step that enumerates all optimal sequences based on the calculated costs. Our algorithm considers structural constraints by estimating MFV for having specific nucleotides at given positions, which allows us to reconstruct ancestral sequences that are energy-wise more fit to both structures compared to previous methods. Let's talk about experiments. For the simulated data, we use 3 randomly designed secondary structures of length 100 for testing. 900 trees are generated for 5 mutation rates from 1% to 20%. And here is the average error percentage of all optimal sequences in a tree. Each column here represents a pair of secondary structures. The first row is for positions in structured regions, and the second row is for unstructured regions. The y-axis is the percentage error, while the x-axis indicates the mutation rate. As you can see, some of the algorithms that we tested against could not finish the run because there were just too many potential sequences to generate for higher mutation rates. The figure shows that Astronomer 2.0 has the lowest average error rate of the 4 algorithms. The results are not comparable when the mutation rate is 20%, since none of the other 3 algorithms could finish the run. Here is the average number of optimal sequences. Each column represents a different pair of signal structures. It shows that the new method consistently produces fewer optimal sequences than the other 3 algorithms, which is good. It means that the approach is able to use more information to narrow down the space of possible ancestors. For biological or real data, we compared Astronomer 2.0 with 1.0 on the FIMP-Traj clan and the GRM clan. Similar to the simulated dataset, the results showed a significant improvement in terms of reducing possible ancestor sequences. We also estimated the results in terms of MFE. For each algorithm, the average harmonic mean of energy is calculated. A smaller number means the sequence is easier to fold into the given structures. In the table, we can see that for both clans, Astronomer 2.0 outperformed Astronomer 1.0 by a considerable margin. To summarize the study, comparing to Astronomer 1.0, 2.0 considers more structural information, applies a more elaborate free energy calculation, and makes use of tree decomposition to partition the cost calculation into subproblems. As a result, it consistently outperformed Astronomer 1.0 as well as classic approaches like Fitch and Sankoff by a considerable margin in terms of error percentage, number of reconstructed sequences, and MFE. Thank you.